Good morning, everyone. My name is Dr. Christine Coons. I'm a physician in internal medicine, and I wanted to start up this discussion this morning, following up on my introduction with my last video, with talking about where most people sit at with their understanding of genetics and biochemistry and things like that. So what I wanted to do is I started out with grabbing a few textbooks. I don't know if people recognize this one. I'm going to pull it into view. Zebra on the front. This is a biology textbook from high school. This was published in 2017. And for the most part, this is going to be the basis of most people's understanding of genetics and genomics. When I went through this textbook, it was echoing a lot of the things I learned back when I graduated high school eons ago. Not much had changed since that time. And a lot of it has to do with history. See, this textbook talks a lot about Mendelian genetics, and a lot of people don't understand what that actually means. So Gregor Mendel is the one considered the father of genetics. That's not contested by anyone, really. It's we use him in biology all the time as a foundation to teach and understand how complex genetics really is. It's the stepping stone. So this is talking about the first steps of our understanding of genetics and genomics in history. So Gregor Mendel passed away in the late 1800s. This is the stories of him being in a monastery and studying the peas. And he was taking certain characteristics and analyzing their inheritance patterns based off of their appearance. And to be honest, Gregor Mendel went through a couple of features of animals to try to figure out which one was actually appropriate. He did at one point decide to try to use bees, and then decided after looking at a couple of these analysis that he's like, nah, we're just going to stick with peas, less bee stings. So he did his studies. They weren't very famous. They weren't very popular. He was a contemporary of Charles Darwin. Of course, Charles Darwin was getting all the attention at the time. So Gregor Mendel passes away and his research kind of fades off into oblivion until it's picked up again in the early 1900s. This is where we get the Punnett square. So in 1906, Reginald Punnett, the Punnett square, wrote his book called Mendelianism. And this became a very popular scientific text and ended up in a lot of schools, a lot of university institutions. So this is pretty much where we are at right now with this textbook in biology. I'm going to get this on the screen. This is what this textbook talks about. This is phase one of our understanding of genetics and genomics, is talking about Gregor Mendel, simple Mendelian genetics, and then this one goes into some brief discussion about more complex inheritance patterns. But in and of itself, that's about it. So I don't know if a lot of you remember going back into high school days of drawing out Punnett squares, talking about inheritance patterns, predictions, and statistics and things like that. But for most people, that's where they stop with their understanding of genetics. That's about 120 years ago. A lot of things have transpired since then about our understanding of where this all happens. I mentioned in the last video that the sex chromosomes themselves weren't discovered until about 1906 by Nettie Stevens. So with this textbook, with that discovery of the sex chromosomes, like I mentioned on the last video, we got really, really excited about chromosomes. Most people don't understand that, or a lot of people don't understand that we have 23 pairs of chromosomes for a total of 46. They always get kind of this look in their face about what does that mean? So we have a total of 46 chromosomes. We have 2 sex chromosomes, but we split them in half to say pairs. Why do we even split them in half to talk about pairs? Well, it has to go back down to the gametes, the sperm and the egg, through a process called meiosis as compared to mitosis. Meiosis basically takes that 46 chromosome set and divides them into 23 in each of the gametes. When conception occurs, those combine and we get a total of 46. So when we talk about these inheritance patterns and draw this Punnett square, that's what a lot of people are talking about. And this is also what the executive order that was released early in Trump's administration is talking about, is they're basing a lot of this on this information back from 120 years ago, because that's the common knowledge in our population as of right now. If we get that, if we do get that. So a lot of the stuff that I want to talk about is more advanced than that. It's taking down layers and steps into more in-depth understanding of genetics and genomics, and how that applies to medicine, how that applies to individuals, and how we can expand and expound our understanding of how this all works. If you go back to the Punnett square in of itself, it's not hard to say, well, based off of sex chromosome analysis, we get 50% male and 50% female. For most people, that's all they want, that's all they need, that's where they're going to stop. But that's not where medicine, science, and even populations exist. Maybe as a general rule, maybe, but that's not, or there's a group of people that we have to account for that need to be included in this group as well. So we have to account for that. So shortly after the sex chromosome discovery and this book being released, we had this massive influx of research going into medicine about genetics and how this works in the human body. We also started looking at individuals who seem to be different than other people. This is where we came in with, oh, we discovered Down syndrome. This is where we discovered Klinefelters. This is all in the early 1900s to mid 1900s. This is where we pieced together Turner syndrome. And what I want to point out is when we look at textbooks, even medical textbooks today, and we talk about Klinefelters and Turners and Down syndrome or Edwards syndrome, we are getting the most prevalent presentations of these conditions. And we have to realize that not everyone has these manifestations clearly evident externally. So the classic, for example, classic of a Turner syndrome individual in short stature, webbed neck, kind of a youthful appearance, pre-pubertal appearance. And that's not the reality. The textbook is only showing one representation of how this appears, and isn't the reality that we're seeing in clinics and in offices. There's a ton of representations and variations of how these manifest from person to person to person. We're just going back to when they were first discovered and first clinically described and carrying that forward. What we're seeing is we're breaking the rules a lot. That actually happens a lot in medicine, where we learn the textbook and then we have to kind of think outside the box of how this all works and pieces together. So when we're going back to Mendelian genetics from our high school years, we're missing a lot of detail, a lot of information, a lot of layers that we need to actually piece together. So that's where a lot of people sit. And what I want to do with the next videos is take this information, start breaking it down into deeper and deeper layers, and go from piece to piece to piece, and show just how amazing and diverse and variable our human existence is in terms of genetics. So until the next time.